Welcome to everyone who is joining us today for the webinar Sequence Once, Query Often. My name is Jessica Vionis, and I'm the Director of Marketing here at Knome. As we're going through the presentation, please feel free to type your questions into the questions pane in GoToWebinar there on the right-hand side. We will answer as many as we can at the end. A short disclaimer slide here, which I'll go ahead and let you read for yourself. Basically, in short, Knome is not a medical provider. And now I'd like to take a moment and introduce Dr. Ben Salisbury, who joined Knome in 2012 as the Senior Vice President of Clinical Products. In that role, he acts as a liaison between marketplace needs and development of the Gnosis platform. Prior to joining Knome, he served in various scientific and management roles at Genalis Pharmaceuticals, Clinical Data, and Transgenomic. I'm sorry, that wasn't Genalis, that was Genesance, my bad. His experience spans several areas of genetic research and its clinical application, including molecular diagnostics, personalized medicine, population and statistical genetics, and software and algorithm development. Dr. Salisbury holds degrees in biology from Yale University and the University of Michigan. And with that, I will hand it over to Dr. Salisbury.
Thank you, Jessica, and welcome everybody. So today's topic is about the ability to take a sequence, generate that in the lab, query it immediately for your initial needs, and then query it later for a refined analysis or for a secondary analysis of some kind. And the presentation is going to be framed by the four educational webinars that Knome has hosted over the last several months. So we'll go into each of these speakers, what they touched on, and bring that into practice in the Gnosis framework, so the software, database, and hardware system developed here at Knome. So first, an overview of what the Gnosis is. This is a system for processing your data, the whole analysis from reads to report. It's a platform for in silico panel development and deployment. That's going to be the main focus of the presentation today. And the system's designed and ideal for clinical research and translational laboratories, supporting a multidisciplinary team of bioinformaticians, lab technologists, geneticists, etc. And the system is designed to be flexible, fast, and secure. And there are multiple configurations. I'll touch on that at the end of the presentation. So focusing in, as I mentioned, on the interpretive side of the process. So after data have been aligned and called and annotated, how do you take the multitude of variants that come out and focus in on the questions at hand? So the concept that we put forth is that of the in silico panel, where these are version controlled and focused at either a very specific indication or for a general purpose. And you can roll many steps into one, many sub-analyses into one, and encapsulating your lab's expertise to make it efficient and then shareable as well. So these panels can go from lab to lab, be modified as needed for their purposes. So Dr. Heidi Ream from Harvard Medical School and the director of the Harvard Partners Laboratory for Molecular Medicine spoke back in June on supporting genomics in the practice of medicine. Really, the focus really was on all aspects of the practice of diagnostic sequencing. She presented on a few diverse disease types, and they spanned the ideas of gene panels, whole exome, and whole genome sequencing. And among the many issues she raised in that whirlwind tour were flexibility in the sequencing process and variant interpretation, the need for knowledge sharing, both at the expert and kind of community level, and the need to address secondary findings. As you know, the American College of Medical Genetics and Genomics almost 2 years ago published practice guidelines around secondary findings, originally labeled as incidental and recently updated and retermed. So I'm going to take us over now to the Gnosis software and show you how we can apply some of these concepts on some existing exomes. So I'm simply switching over to a web browser here, and I'm going to log in. And so my credentials recognize me as both an investigator and a technician. As a technician, that means I have the right to create projects, bring data into the system, do quality control, etc. And as an investigator, I have my geneticist hat on to do interpretation. I'm going to focus in on a couple of projects here. A project is a set of annotated sequences or call sets. This first one, this platinum exomes trio, this includes a son, mother, and father. This is one of the Ceph families collected in Utah many years ago. So there's no particular phenotypic trait that's being analyzed here or expected to find, but it's a versatile set of sequences to try things out with. So here we have a list of the annotations that were applied to this particular project. And having reviewed this, we can now cancel out of this, and I'm just going to open it up. So it's now loading these 3 exomes into memory. And I have a couple of projects already loaded up and ready to go. The first is this secondary findings panel, and another is hypertrophic cardiomyopathy, based on the work done at the Harvard Partners Lab. But before I go into those specific panels, let's see what we're working with. Let's go to our catalog here, and among all these things that are available, I'm going to add in this exome analysis, looking for all rare protein alterations, so everything less than 0.5% frequency in all of the reference populations. So when I add this in, we can see it's a very simple panel with only one query. I just labeled it all. So we're looking for all protein coding genes, substitutions, insertions, and deletions, as simple quality metric filters, and looking for all protein alterations. So in the exons, looking for anything that's non-synonymous, and a broad definition of a splice region carrying out into the introns. And as I mentioned, the 0.5% maximum frequency cutoff in each of these continental and world populations. The nature of this query is that it's in the mode called present in any. So by putting subjects here in this case bin, we're looking for any variant that's found in any one of those individuals. So whether it came from the father or mother, we'll find them. When I run the panel, it now searches across those exomes and finds any site that matches those criteria. And so this site or variant table shows that we found results in a wide variety of genes. Most of these are missense, some splice, potential splice alterations, and we have the genotypes. And many of these genes have been associated with various diseases. Many of them have records at dbSNP. Very few have a record at Cosmic, which makes sense given this is a germline sample. Looking over here to the right, we can see that there are the predicted protein effects of mostly missense, some indels, some broad or narrow splice defining variants. And the SIFT and polyphen predictions, FAST and PhyloP conservation scores, and the frequencies at the Exome Sequencing Project and 1,000 genomes. And all of these are either unobserved in those reference populations or are rare. Those are frequencies, or rather percentages, so those are each well under 1% where observed. And we could work with this large set, but it might take a while to get through 1,700 candidate variants of interest. So we've built these filtering and sorting tools. So we can filter down to based on any of these traits. Basically, these are the columns of the table. So we could focus in for perhaps on the coding effects. We could look at truncating mutations first if we thought those might be a richer source of results. But I'm going to close the filters and just sort. So same concept here. I will choose my first sort by the external classification, and I'll do this descending. So external classification means annotations coming from ClinVar or HGMD, and if we apply this sort, it'll bring to the top all the variants that have one of these external statements in it, and it's sorted such that we have the disease-causing mutations and pathogenic variants using the HGMD and ClinVar terminology, bringing those up to the front. We see a wide variety of traits. Looking back at the gene level information, we can see that some of these are expected to be dominant or recessive. So it's still quite a lot to go through, and the list stops pretty quickly. So we're still left with well over 1,000 variants that have no classification information in them. So let's step back from this full exome and focus in on a particular area of interest. So first, let's run this ACMG secondary findings panel. This is a more complex panel with 5 queries in it that reflect different inheritance patterns. And these are taken from the guidelines as published, listing 56 genes, each with the inheritance mode that's appropriate for them. And so I've set them up accordingly. Recessive or autosomal recessive I broke out into a single variant kind of query, as well as a compound heterozygous search. And those are each structured somewhat differently. Recessive is just looking at a single gene, and I've put the son as the case, so we expect him to have 2 copies of this recessive variant, and the mother and father to both be carriers in an obligate fashion. But we can just go ahead and run this panel, and what it's going to do is execute all 5 queries, and it will give us results that are found in any of them. And out of all those 56 genes, we're left with only a single variant of interest. Everything else has been filtered out as too common, or not a protein alteration, or not found in these genes at all. There's nothing stopping you from doing a broader search. This panel has just been created as a narrow one. And so we can see this variant with a couple different codon positions depending on the transcript being interrogated for the gene MEN1, which is connected with multiple endocrine neoplasia, which is supposed to be a dominant trait. And if we scroll to the right again, we find that this is a variant observed in the literature. HGMD has a record of it with that disease, and they labeled it as a disease-causing mutation. But if we keep looking at this variant, we notice that in fact it has been observed in the background reference population, 1,000 genomes. That's still quite rare at 5/100%. But it's also marked by SIFT and polyphen as likely to be benign or tolerated. So we can take a look at this variant by clicking on the row. So we've gone from the results table into a focused representation of the single variant. And so here we have a somewhat easier to read representation of all of that data. So here we have the gene with its 2 different translations according to the transcripts. And as you can see, we have a lot of hyperlinks within the system. So this would take us out to the UCSC genome browser or to dbSNP, and it would simply open them up in another tab in our browser. So conveniently keeping all of that information together. There's been no internal assessment of this variant yet, so nothing stored on this particular Gnosis machine about that variant. And then there's again the headline result from HGMD. If we review the call sets, we can see that this A variant, which was present in the individual that we marked as the case in the autosomal dominant query, that variant came from the father. Looking at the father and that son, we can see that the allele depth was quite nice. The confidence scores were extremely high. These were passing variants. An interesting thing I'd like to point out is that we have a reference homozygous variant here in the mother. In a traditional VCF file as our input, we wouldn't be able to see that. It would be just a set of question marks, because the standard VCF file only carries variants from the reference sequence. But by employing GVCFs, as we did here, the Gnosis was able to analyze the set of sequences and fill those in by going to the block compressed records in between variants, pointing out the minimum call depth in that region, and of course there's an indicator saying that that's where it came from. Scrolling down, again, there's no internal association yet, but there's that HGMD statement. Again, a disease-causing mutation, and they referred to 3 papers that were relevant to this. An observation in a Danish family, and then some additional studies. If we open up those abstracts right here within the system, we can see here's quite a lot of discussion about that gene. The particular variant itself is not showing up. In the second study, it does. So there's a mention of that variant being almost as stable or only slightly less stable than wild-type menin. So suggesting that perhaps this variant, if it is pathogenic, isn't fully penetrant, or perhaps it only causes some susceptibility or a modification in regards to this disease phenotype. So perhaps this isn't conclusive yet. But scrolling down a bit further, we can review this gene. When you're studying an exome, this is particularly handy, as few people are expert in all 20,000 plus human genomes. And if we want, we can also open this up and read quite a lot of information about that disease phenotype and symptoms, synonyms, and the inheritance pattern, coming from sources such as OMIM, Orphanet, and Decipher via the Human Phenotype Ontology. There are many transcripts for this gene, 2 of which were specifically interrogated in this analysis, due to them being suggested as the clinically most relevant transcripts by sources such as RefSeq gene or the locus reference gene database. And looking at those allele frequencies now, we can see that in fact this variant was absent everywhere in the world, all of these individual populations, except for the northwestern Europeans, where it was observed at over 0.5%. So again, it's probably not a strict sense disease-causing mutation here, although that's still open for further analysis. That observation is, of course, consistent with observing it in a Danish family. If we wanted, we could go search out at PubMed or Google Scholar. If I click the Google Scholar link for the moment, in fact it finds 2 of those papers we were just looking at, and in addition to showing where that variant is in the paper, whether it's in the abstract or in the body. And then it finds an additional paper up here that wasn't listed, that's a review paper pulling in data from those others. Having reviewed all of that, and if you perhaps you likely want to go retrieve some of those papers and look at them if you're really trying to make a final decision about this variant. But you could enter a phenotypic trait, choose your classification. Perhaps it's a VUS or likely pathogenic, or one of these other terms might be relevant. Choose your mode of inheritance. Probably in this case autosomal dominant, since that's the expected behavior in this gene. And you can add comments, so these are kind of internal for the lab, or draft reporting language, and I'll show you soon where those would show up later on. And once you save this into the system, you now have a record that's available for reuse. Anytime you encounter this variant again, it will show up in the variant table, and it will show up here in these annotations. And you can create multiple records for different phenotypes, and you can update these as well. So there's the opportunity to put on a new timestamp or revise that association, fill in some details, etc. So having reviewed all of that, we might decide that this is a relevant variant for reporting. There are two ways to get variants out of the system. One is to export this table in a tab-separated values file format, something suitable for opening up in Excel. There's a lot of delimiting in there if you wanted to parse out data secondarily. Another way to get data out is to use our new reporting feature. This takes all of those variants that were marked for reporting and summarizes them. This is effectively the same information we just reviewed, including records of everything that was done. So the name of the project, information about the sequences in that project, the panel that was run, and a listing of those variants that were chosen, including many others, as well as hyperlinks. So when you save and then export this or download it, your rich text file format includes those hyperlinks, so for easy use outside of the system. So if I save this project, so I'm just saving the progress that we've made so far, and now I'm going to save this report. So this is going to be our ACMG report, and that was on the son in this family, on son number 1, and I'll save that into the system, and that will be available for review later. Now what I'm going to do is take us to the hypertrophic cardiomyopathy panel, and this one, if we take a look here, this was based on the list of genes tested out at the Laboratory for Molecular Medicine. So that link's available to follow. And there were a number of genes specifically tied into HCM as a trait, and they follow 3 different inheritance patterns: X-linked, dominant, and recessive, and autosomal dominant, which, like the ACMG panel, is where most of those genes reside in terms of their inheritance pattern. Similar kinds of cutoffs establish a rare frequency, and in this case, for each of these, I've marked the mother as our case. We could imagine a family history, perhaps some immediate concerns with her, and take the father as a non-carrier, effectively as a control. And the son, we don't know enough about, so we can just leave him alone. And when we run the panel, we again find a single variant. For those of you who have seen me demonstrate previously, such as in the webinar I gave a few months ago, you'll recognize this variant in troponin T2 associated with a variety of cardiomyopathies, and there was, again, a record at HGMD of this as a disease-causing mutation. Having reviewed the data, the conclusion reached was that this was probably just an anecdotal observation in an underrepresented population, namely a study of a few hundred Brazilians. So if we store that record as a VUS now, that would then show up into this table, as I was mentioning. All right, so that's the highlight of running through examples motivated by Dr. Ream's talk on, in this case, it was HCM and the secondary findings. And where I'll take us now to is our another speaker, Dr. Goldstein. So from Duke, and he was focusing in on neurological disease. Specifically neurodevelopmental diseases involved in a variety of different cases in different disease areas. The one I'm going to bring us back to are the epileptic encephalopathies. And he talked about the use of exomes and genomes and made the point that clinical investigators can quickly capitalize on testing existing patient data as new discoveries are made. In other words, you have that exome on file. You only looked at the genes that you knew about previously. Otherwise, things might get lost, or just unable to prioritize them out in the rest of the exome. But when further work is done that reveals that certain genes are of major interest now, you can add those into your query, add them into your panel, add them into your analysis. And as the little snapshot shows here, they had a big study of de novo mutations showing up in cases versus the numbers that you would expect given the nature of the gene, such as the size of the gene. And there were a number of genes that showed up through this analysis as of interest. So taking us back to our platinum trio, let's do something new. Let's, instead of going to the catalog where we don't have something on this, let's make a new panel. And this is going to be testing my typing skills and spelling. But so we were dealing with epileptic encephalopathy. There we go. And now I'm going to even grab that for later use, putting it on my clipboard. We could fill out more information later, but for now, let's just keep going. And we don't have any queries in this panel, so let's add one. And you have choices here. So kind of the first choice you really need to make is what kind of mode you're working in. We could be very specific about whether we're dealing with X-linked genes or autosomal or recessive dominant, etc. But for now, since we're just kind of getting started with this, I think it might be easier just to go with a present in any query. So we'll be able to look at all the genes regardless of where they fall within the genome. And then we could break those out later. So let's just look for all of these epileptic encephalopathy genes. Right now, it's starting off by focusing on the entire exome again by default. But let's choose to look for particular genes and transcripts. So here, we could be putting in gene names directly if you already had a list, or we could also go search by phenotype. So here I put in our trait name again, and when I search, it finds a long list, well, actually a reasonably short list of results, only 24 genes. And there they are, in fact, on the X and also on the autosomes. And they're involved in a variety of traits, many of which have the epileptic encephalopathy term right there in the disease name. The search was actually conducted on disease name, symptoms for the disease, symptoms, even synonyms for the symptoms. And for each of these, we could expand out our view, just taking the first one here on the list, read about it. Again, we could review data about this particular gene. We could look at the transcripts and add the ones that we want, follow links to NCBI or Ensemble if we wanted. We could also just go ahead and add this one in. It makes sense. So we've now added it, taken it from our results and moved it into our selection. And I manually selected that. To save time, we can just add all of the other genes. So let's just add them all, and it's choosing default transcripts for here. We could have been even more generous and basically asked it for all of the translatable transcripts, but for now, this should do. The only problem is when I look down this list, I noticed that we're missing some genes. That is, we're missing genes that came out through the Epi4K study that Dr. Goldstein was involved. So there was the diamine-1 gene and the synapsin-1 gene. And we're not searching by phenotype this time, but by gene symbol. So let's do our search. Looks like I must have mistyped something. Got those two backwards. It alerts us that I mistyped that, which is mighty convenient. And if we try that again, now we get both genes. And they're both automatically added to our selection because I actually named them specifically by name, and I got the spelling right. So now we can apply that in. We've got a query that's going to look across all 26 genes. I haven't restricted this down by frequencies. And we can, let's focus in on one of our patients, and let's make it the son again. And we will run the panel and see what comes out. And what we find are a variety of variants across these genes. We see a lot of them are showing up in both kind of the mother and the father, and that's because if we scroll over to the right, in fact, many of these are common polymorphisms, so they're not going to be, the responsible variants for this. One of them, however, is quite rare at that same kind of number we were looking at before, 0.05%. We could look at that, notice that it's in this potassium ion channel gene, and we could read about it, review it just like we did before. But at this point, I think we've made the point, and we could go ahead and publish this. I have to save the project first. And now we have the opportunity to save this as a draft or final, and obviously I was just getting going. This is just a first draft. Ultimately, I would want to play with that some more, maybe try it out on some other data sets, break those genes down into the appropriate inheritance modes, set frequency cutoffs, etc. Put in our description there, and we can publish. And so when it publishes, it's going to put this in the catalog, and it's also going to lock it. So it's now locked, and I can't edit it, but I could always unlock and work on it some more. But if we go to our catalog and look under drafts, we can see that there it is, available for use. So with all of that, I'm now ready. I'm going to exit this, and while it exits, I'll be back over here at our presentation. So moving on from Dr. Goldstein to Dr. Nislo. So Dr. Nislo is a professor at the University of British Columbia in their School of Pharmaceutical Sciences. So naturally, they have a large interest in pharmacogenetics, pharmacogenomics. The particular themes or areas that he mentioned, we're looking at the genes that are being studied in the eMERGE project, and then a project that they've set up themselves in British Columbia called PRIME, where they are looking to testing out the waters to see how you can go from something that's at this point a little bit academic and really bring it in full-fledged into the clinic, specifically into the pharmacy, working with the pharmacists in British Columbia. And to get going, they're starting with warfarin as one of the most classic examples of pharmacogenetics. But they're not just sequencing those pharmacogenes, as they call them, but rather the whole exome. That way you have more data to fall back on. There may be new discoveries over time, and that way you have them. You just have a lot more flexibility. So as he pointed out, they're creating a common repository of exomes that can be queried indefinitely as more information on pharmacogenes emerges. So let's talk about warfarin. This is a very different story from the ones we were looking at, where we're looking for diagnostic kinds of mutations, those family-specific mutations in patients or research subjects. Instead, here we're looking at the effects of individual variants, typically pretty well characterized variants that have been studied statistically in their relationship to drug dosing and drug outcomes, so safety and efficacy. What I have here is a screenshot from a website called warfarindosing.org, and as you can see, this is very popularly studied at almost 700,000 visitors over the years that this has been up, and their panel of variants of interest has grown over time as well. Their algorithms have gotten more complicated. This whole complex algorithm includes 7 variants across 4 different genes, and all of these clinical traits, one way or another, factor into any kind of suggestions that might come out of this. I'll point out that they have their own cautionary note about this being an investigational device. So how do we approach this with the gnosis? We don't want to look at these genes broadly. That's more information than we need, although you might choose to refer to it. So let's go back to the gnosis, and this time let's look at a full genome. So this is son number 6. He's another member of that 3-generation pedigree. This time we're loading up a whole genome because there are variants that fall outside of your standard targets of these genes. So promoter or regulatory elements can, of course, be far upstream or downstream or even farther afield than that relative to the coding regions of a gene. But so here we have our son 6. We're going to go to our catalog, and choose our warfarin panel, which I've set up to query those 7 variants. So how is that done? So this time, instead of searching by the category of genomic element or by choosing a list of particular gene transcripts, instead I loaded up a bed file, which is a simple file with the chromosomal positions of interest. Those could be broad regions of a chromosome or a gene. In this case, it's pinpoint locations of these 7 variants. And effectively, I turned off all these filters. I just want to see whatever is found in this genome. When I mark him as a case and I run the panel, searches across the genome and very quickly finds the things of interest. And in fact, a couple sites come out that have these external records of relationship to warfarin response or sensitivity. And in fact, these are kind of the most 2 common variants of concern: the CYP2D6 star 2 allele and this VCORC1 promoter variant. If we look at the CYP2D6, we can see this is a very well-studied variant. Many different traits, in particular around warfarin response. We could, of course, link out to the ClinVar and read about anything else that might be there. But meanwhile, back to our results, we can see these heterozygous for this variant and homozygous for this variant. The other 5 we can take to be reference matching. There is another approach, which is to create what we call an always call list, so that when a project is annotated, it will go back to those block records in a GVCF, if you have that provided, and pull out those reference matching genotypes at the other positions. But here we could export these, but right now I'm just going to keep them in memory, and we go back to our slideshow. And what we would do is we simply fill in those appropriate variants for the VCORC1, CYP2, C9, star 2, and fill in the others as wild type, which is what I've done here. And just to point out for the interest here, that VCORC1 promoter variant leads to a lower expression of the drug target. And so therefore patients respond well to a lower dose. So these are labeled as warfarin sensitive, and a lower dose is effective. In regards to CYP2C9, this variant makes the protein a less effective metabolizer of one of the forms of warfarin. So to avoid the bleeding risks of having an overdose, you might go with a lower dose of this drug. So that's an example of something quite different. So then on to our 4th presentation by James Crawford from North Shore LIJ Hospital. This is on Long Island, and he led a CAP study on how early adopters had made the decision to get into next generation sequencing in their clinical labs. And the study was conducted, started the surveying back in 2012, and published a while later after much analysis. And the things that there was a lot of wonderful information in there, and all of these talks are available on our website. I encourage you to go back and look into them if your interest is sparked. But from Dr. Crawford's seminar, he identified 10 common barriers to adoption. And those are also highlighted on Knome's website in our blog. So feel free to read about them over there. But what struck our attention was that among these 10 barriers to adoption, a lot of them revolved around the challenges of informatics and interpretation, so that your existing staff can handle the work without having to do a whole lot of software development or hiring people that might be hard to find. So what I'm also going to focus on something kind of interesting that he also showed, which is 4 pathways that institutions are taking to get into NGS testing. The first one is starting in a lab that's already been doing PCR-based testing. So they may have their Sanger assays for a set of genes, these small gene panels, and when they adopt NGS, they're simply updating the technology, but effectively running the same test, taking advantage of their accumulated knowledge about the relevant variants in these genes. Of course, being NGS, they may well expand those lists out a little bit since it doesn't really add to their costs, their sequencing costs. This is effectively what was done at that Harvard Partners the Partners Healthcare Lab, where we talked about the cardiomyopathy panel there. Then we have pathway 2, which is to take off-the-shelf assays for your NGS platform. So, for instance, the TrueSight panels that are available from Illumina to run on their equipment, and those that includes they have their own cardiomyopathy panel. So just as I had gone through Dr. Ream's lab's list of genes, you could do the same thing with those TrueSight genes, and have the informatics in the Gnosis that would match up to the lab work taken care of by the off-the-shelf assays. So that's pathway 2. Pathway 3 is a little bit more aggressive, or perhaps actually more current, and that is there are a lot of labs that are just getting started, brand new labs, or labs that are going from other testing paradigms altogether and moving into sequencing. And they're acquiring these now much more affordable NGS systems, and they're building custom panels. Typically, these are driven by the requests coming out of the clinicians at their hospitals, the kinds of tests that they would be looking for. And so they're going to build these panels straight out, kind of like I was doing and showing how you could do with building up an epileptic encephalopathies panel. The fourth pathway is also interesting. This is outsourcing. So this could be your standard send out, where a lab redirects the test to a reference lab of choice. Results come back. They might even be rebranded with the local lab's name. But another option is emerging these days, and that is the separation of the lab work from the informatics and interpretation. So once a clinical sequence has been generated, especially at, say, the exome or genome level, and put on file, can't you go back and look at that and run a test that is not based on a new specimen and a new product of wet lab work, but rather use your genome on file as your specimen effectively? So it's a test on the medical record. And while that might sound a little far-fetched, obviously it's something that's of great interest. Once you have those exomes, you want to be able to use them. And also the labs that are going through the interpretive work would like to get paid. So a remarkable new thing is coming out in January. There's a new set of CPT billing codes coming out, and a couple of them are actually framed around the interpretation of existing exome or genome sequence. This is a real, this could really shake things up in an interesting way. If you're interested in hearing more about that and other information about these new next generation sequencing targeted CPT codes, please tune in to next month's Knome educational webinar being delivered by Charles Root, an expert in this coding process. So all of that's really interesting, though, the way things are developing. So having gone through those previous talks, what I want to do is just talk a little bit about where we're going, how you could get going. One is to talk with us afterwards. We are providing this Gnosis system in a variety of ways. One is on installed hardware that Knome is providing. And these are integrated computing and storage clusters that are tailored to an institution's needs. So we have different models depending on the amount of genomics that you're doing and the throughput that you demand and how much storage you need. Then there's the software-only version. So for those institutions that already have a high-performance hardware system that meets our specs, there's the possibility of installing our software and database within your systems. Either way, these first two options, the system are behind your firewalls within the server rooms at your local institution. The third option is hosted, where the Gnome's hardware systems that we own and locally control within our server space we can host a system for you, and we can let you try out data within our secure facility, and you access it remotely. In fact, remotely, just as I've been doing today, accessing one of our systems through a virtual private network, a VPN connection. So with that in mind, we'd like to make an offer to all of you out there today that you get to try out our in silico panels, or perhaps craft some of your own on samples that we already have in-house, or if you would like to provide a few sequences of interest to you, we could bring those onto one of our machines, give you access, walk you through that process. If you're interested, please contact us. You can see Jessica's email below, and of course there are other ways to get to us. And with that, I'd like to thank you for your attention and turn things over to Jessica.
Thank you, Ben, so much for putting together this excellent presentation on practical application of the other webinars we've had this year. A recording of today's webinar, as well as the slides, will be available on our website. I'm going to try and have that up by Monday, so you can check back then. You can also stay up to date on all of our webinars, including the one that Ben mentioned by Charles Root on the new CPT codes, by going to our website, and we hope to have that up in the next week or so, as well as the social media channels listed there. And as Ben announced, don't forget to email me if you're interested in trying out these panels or your own in the Gnosis. All right, so let's get to some of the questions that have come in. I've seen several really good ones. Let's start, Ben, with does the Gnosis offer sequencing, alignment, and variant calling?
Great question. So the presentation today was obviously focused on starting with annotated VCFs. So sequence alignment and calling can in fact be done on the Gnosis. That's a feature that our design concept from the beginning was that needed to be flexible, so that if you have your own favorite aligners and callers, that you can run those, and those may depend on whether you're running a germline sequence or a somatic tumor sample. So to provide that flexibility, the first thing we did was just open up the hardware so that these grid computing systems that we offer can be partitioned so that the Gnosis software is running on one half and third-party tools can be installed and run on the other, including these aligners and callers. The beauty of being able to do that, of course, is that the data are right there. When they come out of that software, they're available to be picked up in our UI and run through on the analysis. Meanwhile, we also have in development the user interface extension that will manage that process, and the different aligners and callers can be added in a modular fashion.
Okay, thank you so much. Another great question that I saw that I know is a question that I've seen in other places is, what are the advantages to doing panels via genome exome sequencing instead of just running like a targeted panel?
Sure. There are of course benefits to a targeted panel. Could be somewhat cheaper to run. Could also have refined, maximized coverage of all of the regions of interest. There's nothing wrong with doing that. More and more, I think we will see over time that labs are choosing to run whole exomes. The enrichment process is getting better and better. Nevertheless, there do tend to be holes. Something like 5% of your exons might not be adequately covered. So there are some drawbacks, but of course the big advantage is that you have access to all those other genes in the future when it turns out those are needed. So especially if you're really in a research mode, You want to maximize the information that you have both today and in the future. So some trade-offs, but you can go either way, and that's part of the beauty of separating the upstream work, so your wet lab process, from the informatics. So the in silico panel could be one-to-one with wet lab panels. So the same genes that are in your enrichment assays are in your queries, but you can also have it so you have a broader sequence on hand, and then do your targeting informatically.
Okay. Moving on to another question. There were several times where you used what we called the gene finder when you were searching for a term to find implicated genes. And the question is, how is that term standardized, and how are the diseases associated with those genes returned standardized? Because obviously there's a lot of different ways you could type something or describe something. How does the Gnosis handle that?
Yeah, that's another great question, and a difficult issue. Specifically, what we chose to do was to bring in information about those gene, disease, symptom, inheritance mode connections through something called the human phenotype ontology, which is primarily a standardization of phenotypic terms. But secondarily, the people who've put that together have drawn connections to elements at OMIM, orphanet, and decipher. So the data is being fed in through those connections established by that third-party group. So when GeneFinder is used to select genes, what it's doing is taking those search terms, and you could string together a collection of terms, and we'll look for them. And it looks for those as substrings within disease names, disease synonyms, symptoms, and even the synonyms of those symptoms. So it looks quite broadly for those. There are other sources where we're thinking of bringing in to allow a broader search for that information. But right now, that already, as you see, that already captures quite a lot.
Okay, thanks. Looking through these, another question that came in is, are all annotations done within the Gnosis, or does someone need to pre-annotate their VCF files?
No, there's no pre-annotation. So the VCF should just have, it only needs to have the sequence data. So what are the variants? What is the genotype? Allelic depths, total read depth, all of that, the quality information that you would want to have access to later, it should be in there. The annotations themselves are applied through the software. So when your set of sequences are loaded into a project, they are, when you launch the annotation process, what it does is it first fills in any missing genotypes, like I discussed, out of the always call list, or variants that are present in one sample and maybe not in the other. So it'll pull those out of the GVCF records. And then it applies the annotations out of our reference data set. I didn't talk about that in this webinar, but we package up every couple of months a new reference data composite that pulls in the most recent versions of all of these sources. The critical point to make there is that we have version control both of the software and the annotation data. So then the annotation is run on that, and it's a fairly rapid process. All of that filling in of genotypes and converting this into an internal format, adding all of those annotations so that they can quickly be used later, only takes about half an hour typically, even for a family. So you can get going quite quickly.
Okay. Today we saw you do some analyses based on VCF files. But one of the questions came in is whether or not Knome has the capability of analyzing aggregates of BAM files to look for copy number variations.
Copy number variants aren't something that we have addressed yet, so the focus has been on things that are well called using the standard calling processes. So the small insertions, deletions, and substitutions. Copy number variation, other structural variants are of course, of great interest and part of our near-term roadmap.
Okay.
Regarding BAM files, I didn't show that, but even if you start with VCFs, BAM files can be connected into those sequences so that the integrated genome viewer can be launched directly out of our software, focus in on those points of interest, useful in QC or in filling in missing data, overall review of data.
Okay. Another question that came in is that our website mentions version 3 of the Gnosis. Can you tell me, what does that mean?
So Knome has been in business since 2007, and over the course of those years, we've gone through quite an evolution, focused on whole genome sequence interpretation for individuals who could afford the rather massive amounts of money that it took to sequence a whole genome back in 2007, 2008. And the software that we wrote then was effectively version 1 for our own internal purposes. Version 2 took this further when we became more of a service company, which we still offer services today. And as part of that service, we used the second generation of our software to provide interactive data sets back to customers so they could launch what were called Knome variants and Knome pathways. To analyze those pre-processed data sets. And so what we see today as Gnosis is the third generation of that technology. The core engine behind that, also known as KGAP, has been transformed again. There's much continuity, of course, but also a lot of rethinking of the purpose, where the primary intention here is to provide systems to customers to use in their own facilities.
I think that was a great description. Thanks, Ben. I think we have time for just one more question, and this seems like a perfect question to end on, and that is, are the reports customizable as needed for someone's own institution?
Yeah, so I would love to say that's all doable within the system, but that's not the state of things today. So the report that I showed is a one-size-fits-all report. You may have noticed that it went on for many pages or screens worth. So the philosophy was to put everything in so that the export file could then be opened up in any word processor, and you could pull out the pieces that you don't want or reorganize them. In the future, we'd love to provide more customization to that process, and more over time. But the focus has been getting the data into your hands for your needs.
All right. Well, with that, we are about out of time. I want to thank
you
you
all for your
again.
question.
Yes, thank you again so much, Ben, and thank you all for coming. Have a great day.